I'm Liam Sanyo for the events team here at scientist.com, and joining us today is Dr. Brian Ogilvie, VP of Scientific Consulting in Scientific Affairs at BioIVT. And without any further ado, I'm very pleased to welcome Dr. Brian Ogilvie. So Brian, thanks so much for joining us today, and the floor is yours whenever you're ready.
Of course, thanks for that. So today I'm just going to cover really an overview of non-CYP-mediated metabolism pathways and strategies for in vitro evaluation. And in the past, we've done a similar type of webinar and then teed it up later for other enzyme systems. So that may be one of the questions, and we will have other webinars in the future. So the outline today will be the introduction, regulatory interest in non-CYP metabolism, reaction phenotyping approaches for non-CYP metabolism, and selected case examples and key takeaway points for future webinars and other things that might be coming. So just as an introduction, to minimize the role of P450s, so CYP enzymes. To start off with, I really want to set the groundwork here, not focusing on clinical drug-drug interactions as much as characterizing metabolic pathways, and some convert to prodrugs, and general approach is to decrease lipophilicity. So when we're talking about CYP metabolism, there's been a lot of work over the years on minimizing the role of P450s through high throughput screening, medicinal chemistry approaches that can be used to produce that metabolism, which is desirable from decreasing drug-drug interaction liability and other aspects related to either rapid metabolism or slow metabolism, depending on the desired outcome for the program. And so if you look at the 2016 issue I've highlighted, this is a special issue on that, talks about a number of different enzyme systems that Foti and Dalby had, talking about UGTs and sulfur transferases. And these two groups are involved in direct conjugation, simply introducing nucleophilic groups, allowing for rapid metabolism. That can result in direct conjugation of these compounds. It does have a drawback, like I mentioned, of rapid metabolism, but not in all cases. And I'll highlight one particular UGT later in the talk where that was definitely not the case. Now for aldehyde oxidase and xanthine oxidase, introducing heterocycles with low electron density, so for instance, pteridines, quinolones, etc., then you can shift metabolism from P450 to aldehyde oxidase and possibly xanthine oxidase. This is generally not desirable because aldehyde oxidase results in very short half-lives, but there are cases where that is desirable characteristics. I do want to mention that we do have a lot of alphabet soup here, so if you're not as well versed in the acronyms, so like CES for carboxylesterases, FMOs, and so on, I will have a slide at the end that highlights all of those. So just try to bear with me, and you'll have these slides at the end to deal with that. So FMO is a flavin monoxidase. It's also an NADPH-dependent enzyme like P450. It's also expressed in the endoplasmic reticulum like P450 enzymes. And by the introduction of soft nucleophiles, so for instance, sulfur nitrogen heteroatoms, you can also increase a shift from P450 to FMO, and this may have some advantages in certain cases. And I won't highlight a case of that in particular today, but I'll overview the general approach for addressing FMO enzymes. And for carboxylesterases, these are essentially just ester moieties that are often used for prodrugs, and that strategy has been employed in a number of cases. Carboxylesterases, there are only two enzymes in humans, a couple of polymorphic forms, and they are expressed mainly in the liver and the intestine. I'll cover a little bit about those later. So for carbonyl reductases, CBRs, and then aldoketoreductases, or the AKRs, introduction of ketones and quinone moieties, you could also potentially include the NQOs in this group. And then for monoamine oxidases, these are mainly amines, like the name implies. These often have problematic PK and other liabilities, but I will mention in brief how to approach those. And then finally, hydrolases. So these are literally hundreds of enzymes. They're often exploited for prodrug activation. And so, for instance, ester amide prodrugs, and I'll cover one example of that in a little bit more detail later. So I do think that in spite of all the work that's been done in medicinal chemistry over the last 20 plus years, reports of P450 decline in drug development may be a little bit early. Granted, this is a look back. This is a 2016 paper, and they're looking at different periods of time from 2006, 2010, 2011, 2015, and then the entire period. But I feel like this is a really great paper from Matt Cerny out of Pfizer from Drug Metabolism Disposition at the same issue I highlighted earlier. He took a look at 125 drugs and binned them between the 2006, like the different timeframes I mentioned. And you can see in the graph the P450 in red, sort of going from 43%, 58%, and then 52%. So I would say, roughly 50 to 55%. It's essentially the same over that time period. If you look at the graph, again, also you see that it kind of goes up and down, but roughly between 50 and 70% for approved drugs over that time period. The percent where P450 was the major medium metabolism, the author noted that although significant, the contribution of non-CYP enzymes in drug metabolism does not appear to have increased dramatically over the last 10 years. And again, this is a look back almost 10 years now. And I'll show you a little bit later some of the work that we've done at BioIVT to update that to some extent. And the change is not significant. I do tend to agree that drugs metabolizing by CYPs seem to be unlikely of little concern anytime soon. So the author notes that although significant, you can look at the non-CYP enzymes, so about 30% between those periods. And drugs metabolizing by CYPs seem unlikely to be of little concern anytime lately. Most of the reaction phenotyping studies that we do focus on P450, and probably next step would be UGTs. And we do have some special cases, and I'll cover those a little bit later. This isn't the only report of this trend, and I'll get to that in just a second, but I want to quantify from this paper the scope of non-CYP metabolism. So if you look at this graph, where Matt excluded the prodrugs, that drops us to 125 total for the entire period. And you'll see that about 18% with no major metabolites, probably more important by transport or just simply renal clearance. But if you look at number one, probably UGTs with about 12%, and then the hydrolases. And I should mention that about 84% of those hydrolases are actually amidase activities, right at 11%, followed by carbonyl reductases down at 2.4%. And then although a lot has been made about aldehyde oxidase in recent years, it's really only about 1%. So just a handful. And I think the reasons for that are pretty clear if you're in drug development. You know that if you miss aldehyde oxidase as being involved, and you assume that it's P450, when you get into your first in human studies, that half-life can be very short. So that can kill the drug almost immediately. So people actually were trying at one point to get to aldehyde oxidase, but I think in many cases it's almost better to have P450 because you have a longer half-life than you would with aldehyde oxidases. And if you go down the list, you'll see the sulfotransferases, very specific cytidine deamidase enzymes, nucleotidases, aldehyde, and alcohol dehydrogenases. And FMO, surprisingly, is way down here at number 9, and then glutathione S transferases at number 10. And later I'll get to kind of my own list of my top 10 and some of the caveats for why these surveys may be limited. So moving on to another paper that was in 2019 out of the University of Rhode Island School of Pharmacy. In this paper, they looked at 250 roughly drugs approved from 2005 to 2015, excluding the prodrugs. But then it did not look at metabolism of the active moiety. But they found that overall, about 50% of the drugs were still metabolized mainly by P450. And then again, the rank order is much higher for non-CYPs with UGTs, coming in at about 40%. Not surprisingly, it also has to do with that old analogy of looking for something under the street light. It's what we know. It's where we look next. And so P450s, probably UGTs following the two most common enzymes to look at, and you're going to find them there. If you don't look at some of these others, you simply won't find them. Other enzymes, these would include categories such as oxidase and dealkylation, acetylation, pyrimidine catabolism, even dehydropeptidases and thymidine phosphatase, among others. And then carboxylesterases, about 10%, probably due mostly to prodrug strategies, along with other esterases, FMOs, MAOs, sulfotransferases, aldehyde oxidase, and then of course the alcohol and aldehyde dehydrogenase, just like in the last study. So this is one way of looking at it. If you look at some of the newer drugs, you will for the last 15 plus years, then the next way to look at it is to look at the top 200 most prescribed drugs during the next period. So in this case, many of these are established drugs. They excluded the ones that have overlapped in the other category that may have been approved and also become part of the top 200. And here again, P450 is about 50%. And UGTs by far the majority of the other major enzymes. And the rank order is slightly different than the newer drugs, but the overall outcome is essentially the same. So between the two papers, they looked at prodrugs, and if you look at the FDA approved prodrugs between 2016, and I'll go to that one here, you can see that of the prodrugs, about 86% were catalyzed by hydrolases. The rest of them made up by kinases, glutathione S transferases, and then some of them were just simply chemical reactions. So established prodrugs, we have hydrolases 78%. FDA approved prodrugs between 2005 and 2016. So later drugs, hydrolases again about 75%. Here, there's more recent strategy with kinases being involved as opposed to demethylases. So quantifying the scope of non-CYP metabolism caveats. I do want to go into the caveats because it's important to understand that when you're looking at what are the most important non-CYP enzymes, first off, there are limitations in data availability. So either mass balance or metabolite ID may be incomplete for some drugs. So for some of you who have looked at a lot of investigator brochures or even the briefing documents for the FDA and NDA package, you'll find there are going to be gaps. That's just part of some programs for certain drugs. When it comes to glucuronides, there's somewhat of a limitation because some glucuronides are generally unstable in feces, mainly because of bacterial beta-glucuronidase, and some are actually unstable in urine, especially in glucuronides. So their contribution to the overall metabolism of an NCE may be underrepresented. In addition, multi-step oxidations that can involve both CYP and non-CYP enzymes, and multiple minor metabolites may be formed by the same enzyme or enzyme groups. So whereas individually, each enzyme may be a minor contributor, collectively they may be a large contributor, but that's underrepresented in these surveys. And then contributions of one enzyme over another may be over or underestimated because there's no corrections for the fraction metabolized being applied in most of these studies. And then finally, because both publications lack data from 2017 to now, and drugs from 2005 to 2016 may represent NCEs that were in discovery 10 to 20 years prior, we may be looking at this in another 5 years, and the look back could be changing. But I don't think that's happening anytime soon. And as I mentioned, we have had a couple of posters. We haven't published this yet. Other than the posters, we did an evaluation of CYP and non-CYP reaction phenotyping study trends from 2000 to 2023, the CRO perspective, and a second, these were both at the Gordon Research Conference and ISSX in in 2024, evaluation of CYP and non-CYP enzyme-based metabolism and preclinical reaction phenotyping, a CRO perspective. So conclusions we had from this. We looked at 353 CYP enzyme evaluations and 141 non-CYP enzyme studies. So again, most people are coming in and asking for, what's the stability in human liver microsomes with NADPH? They're focusing on P450. They may not even include UDPGA as a cofactor for UGTs. They may not even be looking there. So you can see that reflected in these numbers. We had quite a few more CYP evaluations than non-CYP. And it suggests that we had similar contributions of those based as is seen in the approved drugs like in the previous studies. And the contribution of specific CYP and non-CYP enzymes mediating biotransformations also mirrored that of approved drugs. So CYP3A4 followed by CYP2D6, like we've come to expect over this 20-year period, played dominant roles for CYP enzymes where UGT was the most common non-CYP enzyme. But overall, our data suggested there was certainly no shift toward non-CYP enzyme-based metabolism in preclinical drug development. And indeed, the contribution of CYP and non-CYP enzymes mirrored that of approved drugs. Now, with some other modalities that are coming forward that I would sort of classify in the small molecule, you can think of Protacs, for instance. They're certainly not small molecular weight, they may have different types of metabolism, and there are some challenges there. So we're starting to see a few of those and having to look at some of the non-CYP pathways that may be involved. So again, this there's a look back, and things may change in the future. So this is my top 10, and I want to break this down just a little bit with UGTs, number one, and then the hydrolases. And I'm going to call out certain ones, the carboxyl esterases I've already mentioned, and then also within the hydrolases, we've got a number of these that have come out, other esterases and amidase activities. There's a wide array of enzymes, and I've just provided some examples here. And then going into carbonyl reductases, this can include not only microsomal, but also insoluble cytosolic carbonyl reductases. And there are some others that are called short chain dehydrogenases, aldehyde among others, as well as moving on to the FMOs, monoamine oxidases, sulfotransferases, and xanthine or aldehyde oxidase. Finally, we do have requests on occasion for alcohol and aldehyde dehydrogenases, followed by several others. We've had some specific requests for cytidine deamidase, nucleotidases, and glutathione S transferases. And although we haven't published those, so I can't go into some of the specifics, but some of these require tools that are a little bit of literature review to find certain inhibitors of these enzymes, and I'll highlight one approach that we use generally for hydrolases later on. So there is regulatory interest in non-CYP metabolism, and I've included ICH from 2024 as final, so that really overrides the regional guidance, although EMA is coming out with a separate guidance dealing with intestinal metabolism and transport. But you can see that in addition to the previous ones, carboxyl esterases and so on, they've added quite a few in phase 2, specific UGTs, even including sulfotransferases, GSTs, and inositoltransferases. But they do have a statement that is sort of a blanket phrase that means that an enzyme that is estimated to contribute greater than 25% of the total elimination generally needs additional characterization. And they actually put that in terms of both in vitro and clinical. And so if you happen to have, or if you're in an unlucky situation where one of these enzymes that don't have great tools is involved, you have to have a strategy for how to allay those concerns from regulatory bodies. And I'll cover that in a little bit later. So general principles for non-CYP metabolism, choosing the appropriate in vitro test system. Obviously, I'll get into this a little bit more in specific strategies, but one of the first things to consider is what's the best test system? And it's not always hepatocytes. So if you look at metabolic versatility, of course, hepatocytes have more than S9. S9 is simply a combination of microsomes, so the endoplasmic reticulum and cytosol, and then the combination of microsomes and cytosol. And we all have these pooled from many donors, over 200s. But hepatocytes, even if you have a large pool, they may actually confound results in some cases. So you may have fetal cycling. You might have a glucuronide that's formed and then hydrolyzed. You could have low levels of metabolites, and then they can be more complicated, obviously, than subcellular fractions. So suspended, do you want to look at suspended, or do you want to look at plated hepatocytes, or a more complicated system like we have called hepatopac, where you can take that metabolism out to 28 days or so. And in S9, next most complete, you have, as I mentioned before, microsomes and cytosol. You can actually control the activity of cofactor-dependent enzymes, but enzymes can be relatively dilute compared to microsomes. And for microsomes, they often yield metabolites at the highest abundance. You have the highest concentration of the endoplasmic reticulum enzymes. You can control the activity of any cofactor-dependent enzymes, so UDPGA for the UGTs. And in commercially available preparations, I should mention when you're looking at, you're thinking you're focusing only on the endoplasmic reticulum, but unless you use fresh liver to make your microsomes, which is pretty rare, most liver samples are frozen immediately and then made into microsomes later. And during that freezing process, what happens is the outer mitochondrial membrane will actually come off, and that is where monoamine oxidases are. And so you'll have some other mitochondrial enzymes that may also be active. So just one caveat there when you're looking at human liver microsomes. Sorry, I moved ahead. And then commercially available active recombinant enzymes are available for many of these different types of enzymes, and I'll cover that here in a little bit. So choosing the appropriate test system, you want to control metabolic versatility, like I mentioned, with cofactors. So NADPH, that can make the P450s and FMO and microsomes active, but this also goes for carbonyl reductases and some other reductases in microsomes and cytosol. And some of these reductases actually prefer NADH as a cofactor instead of NADPH, and they can, in occasion, use both. So you have to look at both of these cofactors for some of these carbonyl reductases and other reductases. And then UDPGA, as I mentioned before, for UGTs and microsomes. There's now also within the ICH the requirement to use alomethicin or something else that will allow access to the inside of the vesicles, which are microsomes from the endoplasmic reticulum, and then also to include something that will sequester the free fatty acids that can inhibit UDPGA. For sulfaltransferases and cytosol, PAPS is the cofactor, and then for a lot of acidic drugs, acyl-CoA synthetase, you want to have acetyl-CoA, obviously. Those are mitochondrial enzymes. You also have N-acetyltransferases in both mitochondria and cytosol. And an acetyl-methionine is the sometimes referred to as SAMI for methyltransferases in the cytosol and microsomes. And then a lot of enzymes really have no cofactor unless you consider water or oxygen. So you have esterases and microsomes, also in cytosol, epoxide hydrolases and microsomes in cytosol, and then the monoamine oxidases, as I mentioned, in the mitochondria, but also in microsomes. And then you have the aldehyde oxidase, which is essentially only in the cytosol. And this, I should mention, these next tables are from a chapter that I wrote with Andrew Parkinson from 2019. This is in biotransformation. The chapter is biotransformation of xenobiotics, which is in Casaret Duell's Toxicology, the Basic Science of Poisons. So another table from that, choosing an appropriate test system. One of the first questions I typically ask a client or internally is, what is the structure? And from this, you can glean quite a bit of information. This is adopted again from a table I mentioned before. I won't go into everything here except to note that if you look at alkenes, aliphatic alcohols, and amines, of course, P450 can metabolize some of these compounds. And just because a compound has carboxylic acid doesn't mean that P450s can't act on another part of it. But when I have no, it just means that typically, there's always exceptions, can't act directly on carboxylic acid. So you look at possible non-CYP enzymes. You've got these listed here in abbreviated form. And the ones that, so for instance, looking at UGTs, amino acid transferases, and again, some of these didn't meet my top 10 list along with acyl-CoA synthetases. But in all cases, it's kind of a judgment call as to which test system you're using. And you can discern which groups of enzymes may be active by first running in microsomes with and without different cofactors versus cytosol versus mitochondria, or potentially just always going to hepatocytes. So another table, again, looking at the structure, you can go a little bit further. These are all the groups that P450 can act on, but again, I mentioned earlier aromatic aza-heterocycles that tends to shift metabolism toward aldehyde oxidase, but also even UGTs. And some of these enzymes will be present at higher concentrations in extrahepatic tissues. So for instance, where I have human liver microsomes, S9 and cytosol, it's possible that you could also then look at human intestinal microsomes, or S9 or cytosol. And for several of these, especially esterases, possibly even peroxidases, you want to look in plasma, and some will only be present in blood. So you may need to distinguish between those two, and I'll show an example of that toward the end of this presentation. So commercially available enzymes that we have at BioIVT, and there are also some other companies that may have a few others, but we have aldehyde dehydrogenases along with aldehyde oxidase, carboxylesterases, some of the GSTs, MAOs, and then most of the sulfaltransferases and UGTs you could want, especially including UGT 2B10 and 2B17, which I'll cover a little bit later. And then I just want to make sure that when people are looking for commercially available enzymes, whichever they are, make sure that they have activity with appropriate positive control substrates. We've done this as part of our quality control for these enzymes before we release a product. But when you're looking for specific enzymes, a lot of them are available as standards for Western blots, for instance, and they don't say whether or not they have activity. So you would potentially have to order an expensive enzyme and then look at a positive control to see if it actually has adequate activity. So going to general principles again, this is an IQ consortium paper from 2016. It was good then, and I still think it's good now, 9 years later, basically looking at metabolism predicted to be the major route of clearance. So this is either from the structure or preclinical studies, or actually confirmed from radiolabeled human ADME studies later on in development. And if you don't have a major route identified, then you have low risk metabolite victim DDI, and you don't need to investigate much further. In most cases, you will, and then you have to decide, is it NADPH dependent? And if it is, of course, it's not just P450. As I mentioned before, you may need to look at FMO. And if it's not NADPH dependent, then go look at aldehyde oxidases, xanthine oxidase, and also even potentially monoamine oxidase and additional schemes I'll show you here in a minute. But if you do have direct conjugation by UGTs, sulfotransferases, N-acetyltransferases, and so on, then you can look for those in a variety of different phenotyping methods and recombinant enzymes. And if not, then you need to look for other pathways. So these would be the hydrolytic carbonyl esterases, amidases, and other hydrolases. So once you confirm NADPH dependent or not, dependent oxidative metabolism, it doesn't, if it doesn't undergo metabolism in the recombinant P450s, if not, for instance, it's inhibited by 1-ABT, which is a broad spectrum P450 enzyme, although I would caution that it does not inhibit all P450s equally. So for instance, if you're at a relatively low concentration, you may not actually inhibit CYP2C9 until you get to about 1 millimolar. And at that point, it may also inhibit other enzymes. I'm not going to focus on this much because we're focusing on non-CYP enzymes here, but it is important in certain circumstances to look at 1-ABT versus inhibitors of other enzymes. And I'll show you one example of that here in a minute. So does the new chemical energy undergo metabolism, recombinant FMO, or is intrinsic clearance in microsomes substantially inactivated by brief heat treatment in the absence or presence of NADPH? And if yes, you can estimate contribution by individual FMOs, and if not, then FMO is not warranted. So finally, for non-CYP NADPH independent oxidative metabolism looking for clearance in cytosol, can you inhibit by hydrolysine? And I'll mention this a little bit more in the next slide. Raloxifene is also a good inhibitor for aldehyde oxidase and cytosol. And if you do see strong inhibition, then assess contribution of aldehyde oxidase either through additional inhibitors or recombinant enzymes. And then if not, do you see inhibition by allopurinol? So allopurinol inhibits xanthine oxidase, and it's possible that's involved. If not, there may be other enzymes involved. And on the other side, you have primary, secondary, tertiary amines that are metabolized in mitochondrial fraction, are recombinant in MAO. And if yes, then you can look within the mitochondrial fraction. And if you do see inhibition by the two mentioned inhibitors, then there are others for MAO and MAOB. You can assess the contribution of monoamine oxidases. So going back to aldehyde oxidase, I just want to mention that when you're calculating in vitro the fraction metabolized by P450 versus aldehyde oxidase in pooled human hepatocytes, looking only at 1-ABT and hydrolysine, there are a couple of artifacts you might run into. So these are in-house data we generated a while back. We looked at a wide variety of compounds that are P450 substrates as well as aldehyde oxidase substrates. And so the control clearance here is in hepatocytes listed in this column, and then the presence of hydrolysine, you see that dextromethorphan clearance is dramatically decreased. That would give you an apparent FM by aldehyde oxidase of 0.94, which we know, of course, is not the case. Propranolol is also decreased, but you see the same thing with 1-ABT. So almost a complete knockdown or similar knockdown of CYP2D6 activity of dextromethorphan and propranolol. And that's an artifact, because what happens, we found that hydralazine actually inhibits CYP2D6 in hepatocytes with an IC50 of 18 micromolar. It also inhibits potentially CYP1A2 and CYP3A4 to some extent. So if there's a case where you have both aldehyde oxidase and a couple of these CYP enzymes, hydralazine may inhibit those. You may need to also look at raloxifene, which unfortunately also inhibits CYP3A4. So there's no perfect tool, but I think that if you're looking at aldehyde oxidase, you can also then couple that with the recombinants and get to the actual fraction metabolized by a combination of approaches. So this is a UGT reaction phenotyping case example, going back to a paper from Stephen Fowler from 2015. This is one of those papers that when I read it, I really stood up and took note. This was a paper that quoted this phrase, which I think I've heard a number of times: the likelihood of significant drug-drug interaction or polymorphic clearance due to UGT activity, which requires dosage judgment, is often assumed to be low. And I think that's largely because UGTs tend to be high capacity enzymes. In other words, they have a higher Vmax. Their KMs are not typically very low. So people often say, well, there's so many UGTs overlapping substrate specificity, they're much more rapid, etc. But in this compound, when it was developed, it went through a first in human study, and they had one subject. This is, I think, maybe only 19 subjects in this study. They happened to get lucky, I would say, by finding this one subject that had a 136-fold higher dose normalized AUC than the other subjects, and did not form the major metabolite, which was an N-glucuronide. And if you're looking at this structure, if I didn't have this arrow pointing to this nitrogen where the N-glucuronide forms, I think it would be reasonable in preclinical studies to just go through microsomes with an ADPH, and probably has low clearance. It's metabolized to some extent by P450, and so you wouldn't necessarily even look at UGT. Now, as part of the study, 30 other patients were then screened, and they found 2 poor metabolizers, all of which these 3 subjects were actually of African descent. So in human hepatocytes, it produces single glucuronide, as I mentioned, and the kinetics in human liver microsomes actually were biphasic, so suggesting that 2 UGTs were involved, and metabolism by recombinant UGTs was examined. So at this time, they looked at this panel, which, typically these days we'll look at a much larger panel, but this was actually kind of an expanded panel at this time. And they looked, and they found only UGT1A4 could metabolize and form that N-glucuronide. But the next experiment, they actually looked at UGT2B10. So University of Helsinki actually produced the UGT2B10, and they compared it with 1A4 and human liver microsomes versus human intestinal microsomes. You can see the highest rate of formation was in 2B10 and human liver microsomes, which fits with the expression of UGT2B10 in the liver. And again, if you go back to the kinetics, it was biphasic, and we saw that 2 enzymes were involved, UGT1A4 and 2B10. So additional studies showed that UGT2B10 was in fact the high affinity UGT, and results were confirmed with chemical inhibitors dexmedetomidine for 2B10 and hecogeninan, which inhibits 1A4. There's also another paper that highlights UGT2B15 as a potential polymorphic enzyme. So moving on to reaction phenotyping for hydrolases, if you look in the human liver, the human genome, there are over 100 genes that encode serine hydrolases that can be classified as either esterases, amidases, peptidases, proteases, and so on. Other enzymes may have hydrolytic enzymes towards some substrates. So there's at least one case where aldehyde oxidase was actually catalyzing an amide hydrolysis. And that's actually a very interesting paper. It was a kind of a mystery. Jasleen Sodhi is the first author on that paper. And I would recommend looking at this. It's quite a mystery, like I mentioned, in how to come up with the candidate enzyme. It really took a multidisciplinary approach in that paper. So there are some selective inhibitors and recombinant enzymes, but they're lacking for the vast majority of these hydrolases. There are more tools that are becoming slowly available as drugs with some of these enzyme activities move through development, but you have to take, as I mentioned, a multidisciplinary approach. General strategy is to provide evidence that multiple enzymes catalyze the reaction in question, therefore reducing the risk of an effect by a single polymorphism or inhibitor. And I'll highlight one case example that we published on this. Carboxyl esters, I should mention, are an exception to the strategy where we do have recombinant enzymes and good positive control inhibitors and substrates available. So this is a paper that we published in 2012. Interestingly, this drug Zevtera, as it's marketed in Europe, was only approved by FDA last year, as I remember correctly, and it's a prodrug. So it's IV administration, rapid and complete conversion to ceftobiprole through hydrolysis. And the EMA was concerned about which enzyme was involved, and the company also wanted to de-risk the compound. So in vitro strategies were designed to de-risk this aspect of the of the program. So concern, like I mentioned, was raised regarding theoretical risk of altered conversion in special patient populations or from co-administered drugs. We leveraged localization of certain enzymes along with relatively selective inhibitors, and for these hydrolases. The test systems included plasma from healthy and also hepatically impaired neonate and juvenile donors. We also used pooled human erythrocytes, pooled human S9 from liver, kidney, and intestine with and without NADPH. So this was quite a complex study. The chemical inhibitors we looked at are a major part of the study. We have acetylcholine, obviously, for acetylcholinesterases, but you can see all the variety of other ones, including we actually use paraoxon for some of the serine esterases and a number of other enzymes. EDTA was just basically chelates calcium, so it inhibits a number of enzymes, including peroxinases. So hydrolysis was definitely not dependent on NADPH, so we had no evaluation of NADPH-dependent enzymes in this study after the initial experiment. So overview of the results here. If you look at the peroxidase inhibitors, we do see some partial inhibition, especially with EDTA, where you can completely chelate calcium. You can see you don't have complete inhibition, which is good news for this program because this means it's not only peroxidase in plasma that can catalyze this reaction. Otherwise, you would see complete inhibition. So same with some of the reversible inhibitors we looked at. As I mentioned on the last page, you see some slight inhibition, partial inhibition, indicating that none of these inhibitors were completely inhibitory toward one or multiple enzymes. And then really the same with the irreversible inhibitors, where we see essentially no inhibition at all. So from these results, we were able to conclude and additional details from the paper with plasma from different donors that there was no single enzyme that was involved, and that de-risked the compound for any potential polymorphisms or inhibitors. And this is often the strategy you have to take. We've done this with other enzyme systems where we don't have good tools. We don't have a recombinant enzyme. We don't have specific inhibitors. And so you just have to show there's a plurality of enzymes that can be involved in the catalysis of the conversion and show that there's no risk. So with that, I'll get to the key takeaways. Basically, if you're looking at CYP-mediated metabolism of small molecule drug candidates, I do think they will likely dominate for the foreseeable future. And if you look back at the more than 50% of FDA-approved drugs during that period dominated by P450s, UGTs, and some other enzymes may, of course, play a role. But I think CYP enzymes are not going away for the foreseeable future for small molecules, at least. So non-CYP metabolism is important. It can dominate for some drugs. We need a strategy to approach reaction phenotyping based on structure, NADPH dependent, and also inhibition by 1-ABT, et cetera. Now, based on the initial results, whether in hepatocytes or microsomes, other subcellular fractions, the approach may be actually straightforward. So if you see heat inactivation, for instance, you know that you're potentially looking at FMO if it's NADPH dependent. If you know there's hydrolysis and you can show recombinant carboxyl esterases, specific inhibitors can inhibit that. But in some cases, you need to take a broad approach, like I mentioned with the ceftobiprole. And hydrolases, more specific enzymes will require a literature review, and what I would call in-cerebral acid development. There's really no silver bullet for some of these, although I will say AI may be getting us closer to these with structure and knowing which enzymes can catalyze some of these reactions. So I think that in the end, the focus here was to give you an overview of strategies as opposed to individual methods for individual sets of enzymes. We have had other enzymes in the past on UGTs, sulfotransferases, aldehyde oxidase. And so I'd be interested in hearing, now if there's feedback on other types of enzymes people are interested in hearing about in the future.
All right, so first Q&A question of the day, Brian. Will you go into more detail for specific non-CYP enzymes for future webinars?
Yeah, we have some of those from the past, and we can certainly redo those. I think it was worth redoing this overall one to begin with, and then we'll have specific sections. We've done some on UGTs, sulfotransferases, even had one on aldehyde oxidase in the past that we can certainly update. So yeah, good question. We certainly want to keep the interest up here.
Fantastic, great to hear it. All right, can you comment on species differences for carboxylesterases?
Yeah, so that's a great question. So in humans, like I mentioned, you have carboxylesterase 1 and 2. One is expressed predominantly in the liver. There's also one that's expressed in both liver and intestine. But in rats in particular, they have multiple forms, and they also secrete into the plasma. So if you look at plasma studies like I mentioned, one of the strategies was to look at plasma and blood for some of these hydrolases. If you will see a huge difference between rat plasma if it's catalyzed by a carboxylesterase versus human plasma where it is essentially not secreted.
Fantastic. All right, great answer. Okay, why do HLM contain MAO enzymes? I thought they were mitochondrial.
Yeah, so they certainly are expressed in the mitochondria, mainly in the outer mitochondrial membrane. So I mentioned this probably just in passing in the presentation, but most commercial preparations of human liver microsomes are made by first flash freezing the liver and then taking small amounts of that, going through the centrifugation steps to produce the microsomes. So the flash freezing in liquid nitrogen will cause the outer mitochondrial membrane to come off to some extent, and so therefore you'll have MAO in human liver microsomes. So this has a couple of implications. If you're doing a reaction phenotyping study or even just stability testing in human liver microsomes, if MAO can be involved, it could potentially cause the catalysis of that reaction without even having a cofactor present. So that's the main reason there and the implications.
Fantastic. All right. Okay, are there any other examples of UGT polymorphisms like the one covered for UGT 2B10?
Yeah, so Merck actually published on this in 2012. So even before the Stephen Fowler paper, they had a drug MK7246, drug candidate, I should say, and UGT2B17 poor metabolizers had between 25 and 82-fold higher dose normalized AUC compared to extensive metabolizers. So I think that probably ended up in that drug being shelved.
Okay, then. Okay, under screening conditions, what kind of matrix, like microsomes or hepatocytes, should we start to, or we should start to have a clear overview of metabolism?
Yeah, so that's one that even in consulting, I will look at the structure whenever I can and try to make an educated guess, more just from experience, on which system would be appropriate. A good example of that is actually ezetimibe. I don't think it's used very often, but it lowers cholesterol. And if you look in human liver microsomes, you'll see metabolism by an inhibition actually of CYP3A4. But if you look in hepatocytes or if you look in microsomes with UDP-AGA, and you can tell us from the structure, it's got this nice phenolic group on it. And, I'm looking at that and I'm thinking it's going to be immediately conjugated with glucuronic acid. And in fact, that's exactly what happens in hepatocytes and in the clinic. And you'll find that one of the UGTs metabolizes ezetimibe, whereas P450 essentially barely touches it and doesn't inhibit. So that's all to say that the matrix, microsomes, hepatocytes, got to look at the structure and also decide what you want to answer. I mean, are you looking for specifics, specific types of enzymes? If you want to go with what might be most relevant, I would probably start ezetimibe as my example in hepatocytes, knowing what I know about that.
Fantastic. Okay, Brian, can you comment on the variability of the activity of AO in hepatocytes?
Yeah, so aldehyde oxidase, there was a great publication. I can't remember who it was. I want to say it was a Pfizer group published a while back, and there's a huge difference in not only the mRNA expression, but the proteomics, the quantitative proteomics, and the activity, and it varies tremendously based on exposure. I always get these two vices mixed up. I don't know if it's alcohol or if it's smoking, but one of those has a dramatic effect on the activity of aldehyde oxidase. So there can definitely be variability. And I think that's another reason why, not only because of polymorphisms, but because of environmental exposure, to use a very large pool whenever you can for hepatocytes, or even if it's cytosol, if you're just looking at aldehyde oxidase. And of course, the recombinant aldehyde oxidase, um, aldehyde oxidase and xanthine oxidase, all the molybdozymes, as they're called, are very complex. There's another enzyme that has to insert the molybdenum into the enzyme, and so that you don't get the full act. In other words, like you have aldehyde oxidase expressed, but not all of it will be active in the recombinant forms, and that's probably also true to some extent in hepatocytes and human liver cytosol.
Right. Yeah, really great points. It looks like that's all the questions we have, but one maybe just to finish off a forward-looking question, Brian, are there any specific research avenues or new developments that you're especially looking forward to within the next maybe 5 years?
Yeah, I think with a lot of these drugs that are being developed now, like Protax, I mentioned, trying to find more solid analytical methods to analyze those, and just developing strategies in terms of how they're metabolized, that can be difficult. We've also looked at a number of peptides over the years, going beyond small molecules, but even oligonucleotides. So essentially, the FDA looks at oligonucleotides as small molecules. They have a small oligonucleotide guidance document, and they actually refer to the 2020 FDA drug-drug interaction guidance. So I think that that will continue, but I hope that over time, as we get more oligos approved and people continue to realize that they don't have interactions with P450s and other typical enzymes, that we won't have to do much in terms of these in vitro DDI studies that I believe personally are largely unnecessary. But looking more at the metabolism and what happens to them, I think will continue to be important both for peptides, oligos, and even ADCs, and some other more complex modalities that are being produced.
Yeah, really great answer. Well, Brian, thanks so much for your insights today, both in your presentation as well as the Q&A session. It's been a pleasure having you with us.
Yeah, and thanks to the audience and for the questions. I really appreciate that.
Definitely. Big thanks to the audience for participating here.